Today we're going to look at the process of replication. Replication is what happens inside the nucleus of each of your cells and most cells in general when the cell is preparing for some kind of cell division, either meiosis or mitosis. You're aware during those phases that in S of interphase, the chromosomes duplicate so that there are two sets to pass on to the daughter cells, and replication is the process that the DNA undergoes to make those two sets of chromosomes of DNA to be able to pass on to the daughter cells. We're going to have a look at a diagram in just a minute, but off screen, I guess, of the diagram that we're about to look at, we would have an origin of replication. So there'd be a spot somewhere in our diagram that we're about to look at off to the right-hand side where the phosphodiester linkage was nicked, where some of the tension was unwound, and where the helicase enzyme, which we'll look at in a minute, came in and started opening up the hydrogen bonds to open the two strands of the DNA molecule. So the origin of replication, again, is off to the side. Please note, in eukaryotes there are multiple sites of where this happens, multiple origins of replication, and as soon as one area is replicated, it winds back up again because chromosomes are so incredibly huge that the nucleus and quite probably the cell would blow up if all of the nucleus or all of the DNA was unwound at one time. So the second turquoise bullet that you see here in front of you, that's the enzyme that would nick in various locations along the genome one strand of the DNA and allow it to unwind a little bit so that when you pull it, or when it's opened up, you don't end up sort of being all knotted up in the middle of the strand because it's not opening at the end. So we're going to move on to our diagram. And you can see that this is a replication fork. On the right, I have labeled origin of replication off somewhere to the right. On the left is a reminder that the DNA strands are anti-parallel, and you can clearly see 5' to 3' marked on both of the original strands. The original strands in this picture are these nice, lightish blue strands that are labeled A, so that's our original DNA. And again, we can see that these strands are anti-parallel. And what's going to happen is, the direction of replication is indicated by this arrow. So the replication is occurring from the right-hand side of the screen to the left-hand side of the screen. Helicase, which is indicated by the letter D here, and there is a legend underneath for these letters. The helicase enzyme, indicated here as letter D, is going to be opening these hydrogen bonds in this direction as things are proceeding. Now, here is your your new mantra. You need to remember this for the entire unit and certainly until exam time, if not for the rest of your life, which would be good, that new DNA, so I'm not talking about the baby blue DNA or the light blue DNA here, I'm talking about the dark DNA that we're about to build. The new DNA that's being built and using this original DNA as a template is always built in the 5' direction toward the 3' direction. I'm not talking about the direction of the original strand, I'm talking about the direction of the new strand being built. So that is something to keep in mind. If the direction of replication is this way from the right-hand side of the screen to the left-hand side of the screen, then this new strand of DNA that's growing on our top original DNA, this strand is able to build 5' to 3' in the direction that this helicase enzyme is opening up. So it just keeps on going and keeps on going, and this strand will be able to wind back up again as that's happening. On the other side, however, we also need to build our new DNA 5' to 3', and from this, if you have a look at the original strand, 3' on that end on the left side, 5' on this end, that means that we're going to have to build in the direction away from the direction that the helicase enzyme is opening up. So what ends up happening is, as helicase opens up just a little bit of the strand, this red element here, which is an RNA primer, is sent in there and attaches to the bases that are exposed. And the enzyme that allows the growing, so the one that did the growing on this side as well, comes in and starts growing in the direction away from the direction that the helicase is opening. So it builds a little bit and then it hits the fragment that was built before it. So these little fragments are called Okazaki fragments. And the RNA primer in red is what tells the DNA polymerase where to start the next little piece. So if you can visualize, imagine that helicase here is opening up this next little piece. A primer, RNA primer, will come in here, make a big signal, hey, here I am, come and join. DNA polymerase will join in here. That's the enzyme that adds nucleotides. And it will build the new little strand in this direction until we hit the old strand. DNA polymerase cannot join these little strands together, so it takes another enzyme to make a continuous line, and that enzyme is called ligase. Before the ligase comes in, though, we need to get rid of these little RNA primers, because RNA primers are made from RNA, and we don't want RNA stuck in the middle of our new DNA strand. So what happens is that DNA polymerase not only adds new nucleotides, but DNA polymerase also proofreads. And when it comes along and it sees this RNA, it'll basically chop it out, exonuclease it, take it right out, and pop in some of the correct nucleotides, but it can't connect backwards. So it can't connect this piece to the piece before it. So once it's replaced, we send in DNA ligase, and DNA ligase is going to glue all these pieces together so we get a continuous strand. So these little Okazaki fragments are only temporary. They're going to be fixed and set to rights. So a couple of other things. This is the legend, and you can see it kind of corresponds to the letters that are up there. So you can double check what I'm saying to you here about continuous strand. The side that the continuous replication is able to happen on this side is called the leading strand. It's easy on this side. This strand, because you have to kind of work backwards all the time, is called the lagging strand, and that's where the Okazaki fragments are formed. I mentioned helicase already, and the RNA primers that will tell the DNA polymerase where to start. So things that are not mentioned on that diagram. DNA polymerase is the enzyme that's going to add the new nucleotides in. Also, it checks or proofreads, and it can replace the RNA primers with DNA nucleotides. Ligase is like a chemical, an enzyme that's like a chemical glue, can help join the phosphodiester linkages in those free nucleotides and make it a continuous strand after the RNA primers are moved. The other thing not mentioned in this diagram is the single-stranded binding proteins. And the single-stranded binding proteins are going to come in here, and if you, we'll move back up to the diagram, if you have a look, you'll see that helicase has already broken this bond right here. Well, they're almost, they're still quite attracted to each other, and if nothing holds this spot open, these will hydrogen bond back together again. We call that annealing. So the DNA will anneal. We want to prevent that because we want the DNA polymerase to be able to come in and put the nucleotides in for the new strand. So generally we would have some single-stranded binding proteins that would bind on both sides and hold the strands open, prevent them from annealing. And that's what single-stranded binding proteins do. So here's another diagram for us just to have a quick look at. This is a little bit of a different perspective, maybe more of a 3D. We can see the original strand of DNA here is one yellow strand, one blue strand. That's our original DNA. We can see helicase here is a purple X, and it's opening in this direction. So it's going opposite to what we saw in the other diagram. In this case, helicase is opening the strand of original DNA, moving from the left-hand side to the right-hand side of the screen. And we can see that on the top, our original strand is yellow, and it was running 3' on this direction. So we can have our 5' end. Our new strand start continuously, so this is our leading strand, and it's moving, it's being built in this direction. So DNA polymerase would have come in here and added all these nucleotides that are indicated as our sort of new blue strand. And DNA polymerase is this little purple, looks like a little purple eraser. In this diagram, we can see it's indicating that this new blue strand is what we call a daughter strand, and we have single-stranded binding proteins in here that are holding it open. The bottom strand of our original DNA is the blue strand, and we can see that we're going to have an issue here because it's 5' on this end, 3' on this end, and so we're going to have to make our new DNA in this direction, away from the direction of the helicase. And if you have a look here, you can see it's showing an Okazaki fragment here. So it was built, the new piece was built from 5' to 3', and then there's another piece, 5' to 3', and another piece, 5' to 3'. And now you can see ligase is going back through here. DNA polymerase has already pulled out the primers, and ligase is going through and it's going to connect here. And we'll have more ligase that will come in. We've got DNA polymerase here that's still building this strand. So we'll have ligase that comes in and closes that up after the DNA completes and proofreads the new strand. So in the end, we're going to have 2 strands, each made of one original piece of DNA and one new strand of DNA, all wrapped up and ready to pass on to the 2 daughter cells. Be prepared to take some notes and to come to class with some questions.